All right. Thanks a lot, and really thank you for the invitation. It's also nicely dark for the last session, so you can slowly prepare to be done with the science. But I did want to deliver this one story that we have been working on for a very long time now with a lot of collaborators. The few I would like to acknowledge on the first slide, and as I go along, I'll also acknowledge others, but has been the postdoc in our group, Thomas Sokolowski. And this has really been a very kind of engaging and long-term work together with Bill Bialek and Thomas Greger. So let me start by a few thoughts on optimization, because that's what the session is all about. So, I think optimization-based predictions really had a lot of influence and have been present in biology for quite a while. They have been really central in dealing with biological complexity, and I think we can expand on that. And of course, in some sense, those successes and the potential trace back to the fact that basically the evolutionary process itself can be seen as at least adaptive evolution can be seen as an optimization process when, of course, in some abstract space here of genotypes, fitness or selection is acting on traits, and it's kind of moving the population up this fitness landscape. Now, of course, turning this into a predictive framework is difficult because fitness depends on many things, on the environment that we might not be able to mathematically write down. And so what we do either explicitly or conceptually is try to find proxies for fitness, quantitative proxies for fitness that are more mathematically tractable, for which we can write down mathematical systems and kind of turn the problem into an optimization framework. And here I just selected a few such examples where optimization theories, sometimes using very obvious proxies for fitness, have been successful. So the first example is in the world of metabolic fluxes, where one can write down the metabolic reactions in E. coli, and so stoichiometry is known. What's unknown are reaction fluxes through all these various reactions. But one can then postulate that these fluxes should be set in such a way that what is being maximized is the so-called growth flux. That is the proper combination of components that you need to make another bacterium, right? And so this would tie up directly as a proxy for fitness, because that's basically setting the replication rate, right? And so you might want to optimize these fluxes, and this gives you a pretty high dimensional prediction, right, of how these things should be set in a bug, which you might then try to validate experimentally. And this is all about fluxes of matter or metabolites. This is just an example to tie into the work that we have heard about already today. You might really do matter transport in networks such as this Physarum that we were hearing about, and you might think of optimality in that respect, right? So basically moving the matter through tubes or networks of tubes and how the organism should build those. Something that was very close to my heart and very influential to my thinking were the applications of ideas of optimality in neuroscience, where one, in particular in sensory neuroscience, where the design principle is that given some statistics of stimuli that our neural system might want to encode, for instance, images falling onto the retina, those neurons that are doing this encoding should use their limited resources in such a way as to transmit as much information about the stimulus into neural spikes. And of course, then the brain is operating on these spike trains. And there are many beautiful and non-trivial examples in sensory neuroscience of the success of these optimality predictions. And so for lastly, what I really have been devoting a lot of research time into are similar ideas from neuroscience, but this time sort of applied to biochemical reaction networks, in particular to gene regulation, where you might want to think of information channels whereby concentrations of regulatory proteins are affecting the expression states of genes, and this is seen as if you want, as a noisy information transmission channel. And you can formalize this kind of intuition with the quantities of information theory and ask, what should be the properties, what should be the reaction rates, what should be, the kind of binding on and off rates, et cetera, such that this transmission of information, of course, and this is the simplest setup where one input controls one output, but you can generalize it, such that, you know, limited resources, in this case molecules, are best used to transmit, say, cell state or stimulus information for the cells. So this is, of course, now kind of, some of these are poster childs of, you know, of the successes of optimization, but what I wanted to delve right in is that this optimization approaches also pose very, very big methodological and statistical challenges. I think they're interesting. So these are not just technical challenges of how do you write down an objective function, optimize it. They're really important challenges about how do we even deal with these optimization theories, how do we interpret them, and so on. So let me give you a few. One is the one that, you know, one such challenge we had to address in the second part of the talk. So I'll give a concrete example for this biochemical network prediction. But, you know, we were motivated to take these challenges on, and, you know, I hope this kind of opens the discussion that many of you also on the theory end might get involved with. So one thing that you know, really bugged me for a while is that I wanted to at least have the option to formulate a rigorous statistical hypothesis test for optimality. So if you have optimality theory, you have some data, and now you want to test, right, whether the predictions, optimality predictions, really match the data. And what we usually do, and this is actually a beautiful example from neuroscience, from Jan Karklin and Eero Simoncelli, what we usually do in this optimality kind of approach is that we predict certain things. So here, these are receptive fields in the retina and the nonlinearities of these neurons from efficient coding theory, for instance. It's a pretty difficult prediction in this case. They did a great job. And then basically you select examples from receptive fields from the data, you know, something like this, were measured on real neurons. And then, you know, to caricature a bit, we put this side by side and say, see, these look very, very similar, so our optimization works really well. Of course, you can characterize the statistics of these guys and do a more detailed prediction. But I would say, you know, given the data that we have now and optimality theories we are building, this type of comparison is the first step, but is not the last answer, right? About whether is this now confirming an optimization principle or not, right? These kind of visual comparisons. So I think we should do a better job. We should be able to do a better job as a community. Maybe even more relevant is not to ask a yes or a no question, something's optimal or not, but to loosen this criterion and be able to quantify how close to optimality a given system is. This is all, you know, optimality is a point prediction, right? That's a particular state, right? And there is all sorts of discussion whether biological systems can really be taken to that optimal point or somehow they're on the way there, whether evolution could have pulled them all the way there, etc., right? So perhaps a more interesting question is to have a formal answer to how do we talk about closeness to optimality? How do we quantify that? Another interesting point is if we have an assumption about what a system is doing that we can pose as an optimality principle, can we somehow use that to help us infer the parameters of the model? We have seen many biological networks that if you write down will have tens and tens of parameters. Typically, the data is never sufficient to constrain those parameters, all of those parameters. But if you, in addition to data, if you have a mathematical intuition, a formalized intuition about what the system is doing, can you somehow combine that together with the data to improve your inference performance? All right, and lastly, and this is what we will use today, can we use the data to actually, when we do optimality predictions, can we use simultaneously the data to kind of set the constraints of optimality theories or break its degeneracies? So everyone who has done optimality, typically when you optimize something, you optimize it at a given constraint or some number of given constraints. You might not know what the value of those constraints are. You know they exist. So oftentimes the problem is much harder. You would want to test and optimize while at the same time inferring the few constraints from the data that you do not know. So the question is, how would you do that? Or you might have an optimality theory that doesn't give you a unique prediction. It gives you multiple possible predictions that are degenerate. So how do you break the symmetry and say one of them is close to what we observe in nature and the others might not be? So these are some of the challenges. I'll spend the next, I would say, 3 slides to kind of give a direction to how I think one can tackle them, and then I'll delve into an example. And just to emphasize here, I think these issues get really, really hard for complex models. Complex models meaning now optimization in models that have tens of parameters, not if you scan one parameter and find a single optimum, right? And I think they have been actually quite limiting these constraints in many ways. All right, so to give an idea, let me be very explicit about contrasting optimization theories, which are kind of called top-down theory, with kind of general bottom-up data-driven inference. So in an optimization theory, you want to find a model that optimizes some notion of biological function. So typically you start by writing down a utility function that quantifies the performance of a certain system, right? And you also write down a model for how that system might work. Here it's a probabilistic model, and this model has parameters theta. And so optimization is typically independent of the data. It's an ab initio prediction that says, given the utility function, find me parameters theta that maximize the utility, right? That's the sort of plain vanilla optimization, right? Contrast that with inference, so building statistical models from data. So here the goal is to describe the data by sort of a best model in some sense. So you start with the data, you're agnostic of the function of the system. You have here parameterized model, and what you're doing is inference. So you're moving from data to sort of a set of parameters that best explains the data in some sense, maximum likelihood, kind of posterior, maximum a posteriori, whatever you might want to pick. And so, put in another way, on here you have the normative theory, right? It's making predictions about parameters by maximizing the utility. Here you have data analysis or inference. It's again making statements about the parameters, but by starting with the data. So both of these approaches are sort of making statements about, in some high-dimensional space of parameters about where we should be. So if we are optimal, we should be here. If given the data, we are here, right? So both of these approaches are making statements about the space of parameters. And our question has been, and we, with colleagues, with Viktor Mlinarsky and Misha Ohledik, we addressed this in this paper. Don't get scared that it's neuron. It actually could have been applied to any sort of other optimization in biology, but that's the interdisciplinary work. You have to put it somewhere. So our interest was, however, how to interpolate between this extreme that uses no data and just a putative optimization function, and this extreme, which is ignorant of the utility function, uses only the data. Shouldn't there be an interpolation between the two, which might be very, very interesting for biological systems? To give us some example, right? So here you could kind of think of using an optimization theory with a little bit of data if that theory is degenerate or it needs to have its constraints set, right? So to use a little bit of data to break this degeneracy or to fix a few constraints while optimizing many other parameters. And here you could think of a regime where you do the inference, maybe of a high-dimensional problem like learning receptive fields, but theory, the whole optimization theory is used as a regularizer for how to do that best. Okay, so there are interesting cases in between, and we discuss many of them in this paper, kind of corresponding to these various interpolation scenarios. Right, so what would be a statistical framework for combining optimization and inference? And here is the one slide key idea, and then everything can follow from that. So the idea itself is very simple. So one tries to do proper Bayesian inference, but what one does is, of parameters theta, what one does is one encapsulates the optimization theory that's given by this utility function into a prior over the parameter space. And that's a maximum entropy prior in the sense that this gives a distribution over parameters depending on this hyperparameter beta that is most random while achieving a certain average utility. All right. So if you imagine so what is this distribution like? If you kind of take beta to 0, you're getting kind of a flat distribution over the parameter space. If you're taking beta to infinity, you're localizing the parameters at the optimal point. Right. That would be a point optimality prediction without any data. Now, of course, in this Bayesian framework, you're combining this prior with the classical fitting term, which, here is just written out as a product over independent observations. to formulate a posterior over parameters, which now encapsulates both, it takes into account both the data and the utility with this parameter beta, which is saying how important your theory term, if you want, your optimization term is versus your fitting term, right? And now by changing this beta, you can move from sort of an optimization theory, which actually corresponds also, if you think about this, as a low data limit, where the data, as if you had only a few data points, so the prior term is winning, to the sort of weak beta limit, which is close to statistical inference, where the data term is winning, and maybe the theory is just regularizing your inference. And then you can, starting from there, you can attempt to answer all those statistical challenges that I was posing in the beginning. And today what we'll do, and I'm flipping now to the concrete example, so we'll attempt to derive a complex biochemical network, sort of ab initio, and we'll use this strong beta regime here to break the degeneracy of its predictions. And of course, now I need to make this a bit more concrete. All right, so switching gears now. We are thinking of building an optimization theory for spatial patterning. And in essence, this is trying to address the question how cells in a multicell organism, even though they have identical genetic material, how can they reproducibly assume different cell fates which are appropriate to their position in the body? And I'm sure many of you are aware of how this of the framework here. But what I like to think about is to contrast this problem with the problem of kind of finding where you are on the surface of the Earth by radio triangulation, which is what the classical GPS does. And in a biological system, the cells here in a classical French flag model arranged in a one-dimensional lattice, cells determining where they are, not through radio signals, but through chemical signals. So you can think of this as chemical GPS. And the conceptual picture here was that what gets established along the spatial axis of this toy model embryo are concentration gradients. Here there is one of them, this exponential decaying profile, and then cells can read out local concentrations and thereby determine where they are and which one of the three colors they would like to become, or they should become. And so the concept has been that these gradients encode positional information, and cells can read this positional information out. And this concept of positional information has been very influential in developmental biology. Now, one might immediately ask what kind of spatial profile, what kind of shape would provide the maximal positional information? And that was indeed a question that was asked very early on. And you might say, well, it's just linear. And that's actually the case if the noise is constant along the length, but oftentimes it is not. But the question gets much more complicated if you, instead of one morphogen gradient with constant noise, you consider what would be the optimal shapes if there is multiple gradients and their noise changes as a function of position and so on. So what would be their optimal shapes to provide maximal positional information? And so with our colleagues and longtime friends, so with Thomas Greger in this particular case, we started thinking about this in the context of the developing fly embryo. Again, this is a very early stage, several hours after fertilization, where there is no morphological changes yet, and cells are arranged on this 0.5 millimeter ellipsoid. But soon after that, the first changes start happening very reproducibly, like this invagination here of cephalic furrow. And the way cells decide to do that action at that right reproducible spot is that, as you know, is that before that, within these cells, chemical gradients are set up to provide the positional information we just talked about. Actually, they provide it through a very complicated interacting network. It starts with the mother breaking the symmetry and depositing signaling gradients. So this is a stain of a bicoid. It's one that goes from anterior to the posterior end. And these morphogen gradients then drive the next layer of genes, which are called the gap genes. And sort of there is a cascade by which this precise patterning is established. And looking at this, one starts to wonder that even though the biological concept of positional information is simple, the answers to questions such as how much positional information is there in these patterns, and there's more than just these ones. This is sort of a small selection of them, and there is more genes that you can stain. So how much positional information there are in these patterns, and what patterns would maximize this information? I mean, these kind of quantitative questions have actually remained unanswered for quite a long time. So what I'll focus on in the next slides is this particular stage of gap genes, of which there are 4. And here is, I mean, Thomas and his colleagues, and so this is reported in a paper by Julien Dubuis, but many others too, have been very careful in measuring these 4 gap genes at various points in time. So here you're seeing just one snapshot in the fruit fly embryo from anterior to posterior. These are 4 colored curves which correspond to these 4 gap genes: Knurps, Hunchback Giant, and Krupel, here denoted, with their variability across many embryos, which contains both extrinsic variation and the intrinsic noise. And so our idea was to make this concept of positional information really a mathematical quantity that you can estimate from data in a way that's independent of the organism. Okay, so this would be a good approach also for other patterning systems. And so the key intuition here is to make really precise the notion that these concentrations encode for position. And in particular, you should think of these concentrations as really stochastic variables. So here what I'm showing is average over many embryos and their variability. But for a cell sitting somewhere in this embryo, of course, what that cell can measure are fluctuating concentrations of these 4 numbers. And in particular, one could think of this combination, Krupel being 0, Knurps being 0, Giant being 0.6, and Hunchback 0.8, as a combinatorial code for being at this particular position. Right? And some other code would correspond to some other position, etc. Okay? So to mathematically encapsulate this notion of a code, in particular combinatorial code, for this 1D variable, which is great. So we now have on x-axis, we have x. So somebody was asking what we should put on the axis, right? So that's nice. So to encapsulate this notion mathematically, right? So our proposal was to really identify this concept of developmental biology of positional information with a quantity from information theory, which is mutual information between those 4 random variables and position. And that's now something you can compute from the data and you can reason about, because we have this quantity has many properties that we understand. Okay. So so this is published work, and I'll just go on this one single slide through some conclusions that we could make by estimating this positional information from data according to this methodology. So first of all, we concluded that each of these gap genes individually, so let's say Hunchback, carries more than 1 bit of information, indeed more closely to 2. That was quite a shift because in the community, gap genes were thought to define expression domains. In particular, Hunchback, this guy, was thought to split the embryo into front half and back half. And if it were only that, then its capacity would be limited to 1 bit. So 2 bits means it can provide much more information to differentiate the position than if it only were doing a front-back split. So the gap genes are not binary switches. Together, 4 gap genes jointly encode about 4.3 bits of information, so not 4 times 2, so not 8, right? So the gap genes are quite redundant in encoding the information. Nevertheless, 4.3 bits, one can show, implies that each nucleus along this axis could, in principle, read out where it is with a positional error that's about 1.5% of the egg length, which is very precise. In particular, it's precise because if you look at the nuclear, so cellular scale along this axis, you basically have about 60 nuclei along this axis, which is in close correspondence, right, to the precision that you actually have, right? So you can read out enough, precisely enough to be able to differentiate yourself from the nucleus next door with a very small error rate, right? So then the next thing is that one can measure downstream events downstream of the gap genes and ask how precisely do they happen. And you will find that these, for instance, the patterns of pair rule stripes, they happen roughly with the same precision of 1.5 or 1%, including the cephalic furrow invagination. This this positional error is nearly uniform along the x-axis, so no matter where you are, you have this capability of reading out your position as precisely. And this actually provides an empirical signature that an encoding of positional information might be optimal. So the argument is not completely straightforward, but one can make it. So this has been published. And also, there are signatures that the same gap gene concentrations are also read out by subsequent patterning genes in a fashion that's consistent with optimality predictions. All right, so that's if you estimate empirically positional information from data. So what I want to do now is I want to turn things around. And instead of doing this empirically and checking for signs of optimality, we'll try to assume that positional information is being maximized in a pretty complex biochemical reaction model, and then try to derive, by maximizing positional information, try to derive how this pattern should look like and what the gap gene network should be. All right, so here I really need to acknowledge, very difficult task that Thomas Sokolowski had, because what we ended up doing, or what he ended up doing, is really setting up a spatially resolved stochastic dynamical systems model of interacting gap genes, sort of putting discrete nuclei along the x-axis, and then assuming that what these cells in the embryo are receiving are the maternal morphogen gradients by the mother. So this we know from the experiment. What happens in each nucleus along the axis is that you have, and I'll show this on the next slide, you have these 4 gap genes that can couple to the maternal gradients. They can also cross-regulate themselves in an arbitrary way that we can parameterize. One is modeling the expression of these 4 gap genes, the degradation, even the fact that these nuclei are dividing, so they change in number. And one is computing in a Langevin framework the gene expression noise due to the fact that all of these reactions are happening at finite copy number. So this is because the morphogens are there at finite copy number, because the gap genes are expressed at finite copy number, and you propagate all the noises correctly. Okay. So it's a very big and bulky, biophysically semi-accurate model of patterning. And the traditional way, would be to try to fit its parameters. And this thing has about 50 parameters that determine this cross-regulation between gap genes, how they couple to morphogens, and the diffusion constant of gap gene products, and so on. So one could try to fit this model, but what we will choose instead is to, for each value of the parameters, compute the positional information and then put everything into a big optimization loop, if you want, to maximize the positional information and ask what is the optimal network for encoding positional information. Yes?
Is it prior or after cellularization?
So what we are
doing already cells, or
no. So we are thinking. So cellularization will happen. At approximately. This is nuclear cycle 14, roughly 40 minutes in it. So that's when it happens exactly. We have nuclei, so syncytium. Yes, exactly. And then this is sort of the diffusion that can still take place of gap gene product. Right, so one more thing to say, right? So what limits the information in this case, right? So the information is limited by resource constraint, basically by the fact that when you express your mRNA in protein, there are sort of limited rates of how many of these copy numbers for these products you can make per unit time, which also set the maximal number per nucleus that you could have, right? And so for us, the degradation times and these expression rates are coming from, from trying to match to the data that in Thomas's lab was being estimated, right? So how many maximal number of mRNA per nucleus and how much is the maximal number of protein per nucleus. So that gives the constraint because the copy number set the level of noise, and the noise obviously limits positional information. Okay, so constraint in this case comes naturally. All right, so why, where do these parameters go? So just to give you sort of an account, right? So you have 4 gap genes that can interact in some arbitrary fashion. The way we parameterize this, and I don't go into detail, is sort of there is 2 parameters per every arrow. They can also self-regulate. And then the 3 maternal gradients can drive each of these 4 genes again, individually with individual parameters, right? And so what we want to do is really to predict both the parameters and the topology, and the number of parameters clearly scales with sort of the number of gap genes, squared. Okay, so here is when I look back at these early comments that I was making about being careful about optimization, right? So we are talking here about optimizing in roughly 50-dimensional space. It takes a long time because for every parameter set, you fully evolve this stochastic, system forward in time up to the, time point where we compute the position information. And even though Tomek wrote the code in C, right, each iteration, each evolution of this model is taking time. So if you do one optimization run, this takes several days on the cluster, right? So we are limited. And what we want to ensure is that when we optimize the position information, if there are multiple solutions in the parameter space that have high position information, we want to somehow find the one or check whether one exists that's close to the wild type. So that's our first task. And the way we do it is that we optimize an objective function, which is basically the logarithm of what I showed you in your Bayesian inference. So there is an optimization term. So you want to maximize position information. And there is a prefactor beta that corresponds to this beta in the prior. And there is another term that you could think of a pulling force that pulls your solutions towards the pattern that you measure in the data. Okay? Now, of course, it depends very much. So this is the optimization term, and this is the fitting term, which is basically the log likelihood. And this trade-off depends on this parameter beta, right? And so the way we went about doing this is we ran this optimization in different beta regimes. So you can make beta very weak, basically setting this to 0, and then you're doing pure fitting. This you do because you want to ensure that your model is actually rich enough that it can even reproduce the observed patterns. Okay, that's just to ensure that your model class is sufficiently rich that you could find a solution that matches the data, at least approximately, right? Then you forget about that, and then you set beta to a very strong value. So now the optimization term is dominant, and this is a weak pulling force that's pulling your stochastic optimization towards the data. So this is because if there are multiple degenerate solutions, you want during your stochastic optimization to be guided towards the data and find if there is an optimum there close to the data. And then you can go to infinite beta limit, so you forget this term, to verify that this optimum that you find is actually an optimum, right? You can get the derivatives and so on, right? And also to find potentially other solutions that are optimal but are not like data, because that's also something that we are interested in, okay? So this beta helps you navigate this high-dimensional space during your stochastic optimization. Okay, so let me give you first this, a feel for the results in this high beta limit. So we optimize but slightly pull toward the data. So here is sort of information during one single stochastic optimization run. And if you track the solutions as you proceed with the optimization, of course, you start with some random parameters. There is 0 position information, but you just by chance get flat profiles. And as you go to point number 2, you see the first split of the embryo in 2 parts. You then see this kind of structure emerging that's slightly more complex. The genes also, as I'll show, start cross-interacting with each other. And at the end of this run, and of course that's the one solution that I chose because it will have a nice match to the measured one. So at the end, we find this particular solution in this run that has about 4.2 bits of position information. And this is the one that you should be comparing also in terms of value to the measured gap gene profiles, which are here. And now I'm committing exactly the same sin that I was criticizing ourselves for. I'm trying to convince you by comparing you two pictures side by side and saying, see, they look similar, don't they? Right, so, but just to mention a few things though, I mean, there is many things you can explore about these optimal solutions, but these are the few I would like to really highlight. Right, so there is, one can verify there is the information maximizing solution close to the wild type pattern. There are mismatches which we cannot get even when we fit, okay? So this nearly linear ramp in our model is very hard to achieve, and these little blips that are there, we also cannot reproduce, and I think that's because our parameterization of regulatory functions is not powerful enough to get this. So there are things which we believe due to parameterization we could not replicate unless we made something much more complicated. it's very interesting to see, right? This is kind of a nearly mechanistic model. It's very interesting to ask simply, how is this precision achieved, which was a crucial developmental question in this system, right? How is it that we get these gap gene profiles reproducible to within the same precision as in the wild type? And so there is no black magic, right? It really results from various averaging mechanisms done appropriately. So there is temporal averaging in this system. There is averaging through diffusion, which is very important, actually, spatial averaging due to diffusion. All of it combined, gives you something that matches the data not only in the mean, but also in the variance of the profiles. I would like to also emphasize one more thing. So if you did classical fitting, right, you'd be optimizing some sort of a chi-square or maximum or likelihood objective function, which is statistical in nature, doesn't have a clear interpretation. So here when you optimize something that's actually a measurable phenotype, right? So this is where optimization brings you to 4.2 bits. This has a meaning, okay, this number, and you can really compare it to something you estimate from data directly, which is 4.3, right? There is no reason in particular these numbers must be close, right? It could be that the best optimal solution would be at 5 bits or 7, I don't know, right? So it's nice to have this interpretable utility function as opposed to fitting, statistical utility function. And last point I want to make here is that if you actually do the pure fitting, which we can do, we can just take this same parametric class of models and do fitting with no optimization, and you do multiple runs, because that's also a fitting-wise degenerate problem. So there is many ways to set the parameters to make your pattern. The information that fits get, which usually match the data slightly better in terms of shape, but they have a lower information than the optimized solution. And it's interesting to think why, right? Because you reproduce the patterns if you fit even more closely than the optimal solution. And the reason is that when you optimize for information, your utility function is taking into account the functional consequences of noise, right? Positional information is high not only if the patterns are right, but also if the noise, right, this variability can be low in the correct places, right? And so when you optimize, gap genes, interactions, and so on are tuned such that you can actually get higher information solutions. All right, so you can also open the box and ask, how do the gap genes interact? So one way to check is to compare with what people have painstakingly assembled in terms of the interaction network by doing genetic interventions and mutations. So this is from a review by Yogi Yeager, the known interacting network where Hunchback and NURBS, Jan and Krupel have very strong repressive cross interactions, and then there is sort of weaker set of interactions and some self-activation. And if you look at the optimal solution that I showed, well, it recapitulates correctly this pattern of strong cross repression. It gets some of these weaker interactions right. It doesn't get all the self-activation right. I'm not sure how well that's actually constrained by the optimization principle, but certainly the match is good, although caveats, right? I mean, these are actual numbers in the optimization, so we have to threshold them to plot them. And it's also, I think there is no firm agreement on some of these weak interactions either, even in developmental biology field. All right, so now let me end by putting forth something that really has occupied us longer than just the first optimization run. All right, and that's kind of making a step and releasing this pulling force that's pulling you close to data and just optimizing, okay, with really ab initio. So no force that's trying to match the data in any way, okay. And what you observe is that what you find is really an ensemble of nearly degenerate solutions. They all reach about the same information. So actually here I plot them in this particular way. You'll see why. So this is the information axis. This is sort of, there's many dots here. Each dot is one optimal solution, one optimized solution. That's the median of the optimal ensemble. So this is after many optimization runs. And this is what I'll show you on the next slide, is what we call the random ensemble, right? So where we pick parameter values randomly without any optimization and just compute what would be the expectation. And this is sort of various issues how to do that. But what you see here is that, if you do optimization multiple times, there is some spread in information that you get. You do not get above 5 bits, so it's mainly the bulk is between 4 and 5, and there is different patterns in there, okay? What you find is that while you optimize for information alone, right, as expected perhaps, this same ensemble, which I basically chunk into 3 slots by 3 colors, so lower information, middle, and high. What you find is, of course, that high information will correlate with low positional error. That would be expected. These two quantities are related. But there is also some more unexpected things. For instance, the fact that, you know, being high information means your gene expression profiles are making many slopes. Okay, slope means in those profiles, you know, a transition from low expression to high expression or vice versa. And so you clearly also see here that higher information means on average you're making more slopes. So you want the slopes to encode the positional information. And again, that's the random ensemble. It also, you know, optimization also goes together with a very uniform allocation of resources per gap gene. So you have 4 gap genes, and you know how much each is expressed, you can think of as a cost, right? And so the CV, so the coefficient of variation of how much you express each one of them is quite low in this example. So you know every one of them tries to be somehow active. Surprisingly, we also find this, that higher information solutions, so these are these colors here, and this is the information, tend to correlate with more stability of the pattern. So we have a full dynamical system, right? We evolve it forward in time, we look at the snapshot, and it has high position information, that's what we optimize, but we can then ask, because it's a dynamical system, is that a stable solution? So, you know, if I evolve it further in time, will these patterns just go up and down? Did I catch the system in a transient moment, or is it sort of close to, you know, steady state, okay? So if you optimize just for information, you see that in this ensemble, higher information solutions are actually correlated with low, I mean, these are temporal derivatives, right, of gene expression patterns, right? So they go hand in hand with stable solutions. Okay, this is a complicated plot, so I'll just, but it's kind of nice. You can just relax and look at the colors. So this is now, again, trying to relate this notion of cost of expression and information. So what we have done here is we have, you know, this is my random ensemble. So we just randomly pick this 50, within this 50-dimensional parameter space. Each dot is a particular solution for which you can assess the information and the resource cost. By resource cost, I mean how much are the gap genes expressed, okay? It's just a summed expression over distance. It has this very interesting structure because these are parameters. Here you express 0 genes, and here you kind of express on average 1, and so on, right? So these are strongly nonlinear. So you might pick parameters where 1 gene is just on and the others are just off, right? So this is the random ensemble. And then what I was showing you before that's optimal are these red points here. These are what I call a standard resource cost bound because it's matching the wild type. So optimization is taking stuff from here and driving it all the way up to here, right? And so here you see the marginal histograms of these things. So optimal solutions are in this cloud. You can then do stuff such as restrict the amount of expression that you allow. And you see, and these are now optimized at the lower resource cost, right? So you can spend less expression and even less, right? So this corresponds to resource cost. So you can kind of see, right, that even in the random ensemble, you have this shape. Optimization has just pulled stuff out, but you still have this kind of front, right, which is trading off information for cost. And if you release all the resource constraints, it's interesting to see that you do not go much further higher in information. So this is without any constraint. So you can collapse this right now onto a single plot. So these points correspond to these constraints. These are the medians and top and bottom 90 quantiles. The red stuff is what I was showing you. So these are wild type and optimal ensemble. And this is without any constraint. So you see this sort of saturation where even if you release all the constraints, you actually don't reach above 5 bits of position information. Right, you might, and since these are all completely without any pulling force towards the wild type, you might compare these domains. So this is hunchback, this is space, hunchback, giant, crupel, knurps, where in the, you know, in the wild type where the domains are. And these are some example solutions from various ensembles that you see are not exactly the same, but they're recapitulating the domains, right, of the wild type of 4 genes, right? So where they are expressed. So selected from different ensembles that have compatible resource constraints. All right, so mechanistic limits are in this case, right, are limiting your information to less than 5 bits, so this is the green stuff, but wild-type-like solutions, which are the red ensemble, can be achieved with actually much less expression, so at a lower cost. But if you start tightening the cost, you really start dropping. All right, so I'm nearing the end. I have one more example. So I think these optimality predictions are non-trivial in the sense that you can do the following trick. There is the one parameter I haven't touched yet, which is the diffusion of the gap gene protein. So we have before, in what I was showing, we fixed it to some value and optimized all the other parameters. So the value was about here. I think this was at 0.5 micron square per second. And now what you can do is you can fix it to other values and re-optimize the information. And what you'll find is that if you make diffusion constant too large, even if you re-optimize everything else, your information is dropping quite rapidly. And if you make it too small, it's also dropping. So basically, even if you optimize everything together with the diffusion constant, you will be in this ballpark that we selected. And right, so this is sort of a ballpark which is probably plausible given some other proteins that are diffusing in the fruit fly, like some of the experiments that Thomas has done on the diffusion constant of bicoid. Right, so this is what I said. What's also interesting is if you actually look at how these displaced solutions look like, they just look qualitatively so in disagreement with the fly. So if you make diffusion constant really lower, you will get more boundaries. They're too flat at the top, and the profiles are very steep, and actually that doesn't get you enough information. There is a drop. If you make the diffusion constant too large, you get these smooth profiles. The noise actually goes down because diffusion constant helps average out the noise. But even if you can perfectly arrange them, you're still limited in how much information you get. Right? All right, so I need to wrap up. I knew I had to put this on since Bill was making statements about this. It's like, arguing with your parents, right? But I put it in quotation marks because, numerical optimization here is not evolutionary adaptation. That's very clear. But it could reveal limits and trade-offs that also adaptive evolution might face, because these are sort of biophysical limits, if you want, or resource limits. I think there has been some very interesting work that I haven't spoken about, where this type of optimization could be formally linked to concepts of evolutionary theory. In particular, if you look at this random ensemble and optimized ensemble, you might be able to bound how much information would have to be encoded in the genome in order to specify the optimal solutions. And that's kind of an interesting way to think about it. So the way I imagine its random ensemble is somewhere here on this axis. Our numerical optimization can tell us what's possible by constructing this optimal ensemble. And of course, selection, which has other limits that our computers don't have, is acting to generate the wild type here, and in principle could generate sort of other diversity, maybe if you look at related fly species and so on. And so one can be quantitative about these distances and the strength, how far selection can pull you against biophysical constraints. So that's something very interesting that we are looking at. Are these other solutions, degenerate solutions, maybe realized not in melanogaster, but in any of the relatives? That would be a fantastic question. I don't know the answer to that. And I think what I haven't shown you is that once you have this optimization framework, you can actually explore questions of evolutionary necessity and sufficiency. You can take one gap gene out, as if I were doing a mutation, erase one gap gene, but re-optimize everything else. And really ask, what does the fly need that fourth gap gene? And what would it do if it had 5 under the same resource constraints? So we have actually done these things, and I don't have time to show. And we have some putative answers for, what is the functional role. And all I can say is that this is not trivial at all. Right? This system has so many moving parts and redundant parts that depend on each other that you can construct, I think, maybe a rational story, what's coming out after you have re-optimized everything. But many of these I would not have guessed, even though I spent some time looking at this fly solution. So I think there is something very interesting. All right, so to conclude, I think this was quite a long journey to take a biological concept, kind of a word-like concept, and to change it into a mathematical quantity that's actually system independent, so it only depends on these profiles. You can estimate this in other organisms quantitatively from data. You can then try to elevate that to a utility function of an optimization theory and try to derive some first principles predictions in what is a mechanistic model. So you might try to interpret the terms and so on. I think there is very exciting connections to evolution where this utility function is elevated further as being a proxy for fitness. And you can discuss these concepts that have been in the evolutionary field for decades, and there is a lot of very good discussion, but here you can explore them quantitatively in a mechanistic model, things like historical contingency and, is it tinkering versus optimization and developmental constraints. And last but not least, I would really emphasize this. I think these large-scale optimization approaches are raising very new and interesting, really fundamental statistical questions. How do we combine inference with optimization? How do we interpret whether optimal solutions are trivial or not? I mean, this is not restricted to the fly, right? There's people who do machine learning models of the visual stream and are making statements that the neurons that you observe in these machine learning models correspond to what the brain is doing and so on. And the question is, how do you make that comparison rigorous, right? So I think there is a lot to do here. And of course, what are the relevant constraints, right? Maybe we don't know them all. And if we knew some other constraints, the degeneracy would be lifted. So how do we discover those? All right, so thanks, and sorry for going over time. You've been standing there for a long time. Thank you.